Hello, so today we are going to study a new lesson number 11, cell structure and microorganisms. So let's start with the cell. We have learned that cellular organization is the primary characteristic of all living organisms and that the cell is the fundamental structural and functional unit of living organisms. So our body is completely made up of cells, small cell structures. Let us see the flow chart. Now you can see there are some empty boxes over there. So let us fill up the boxes first. The second box is words. Third box is sentences. Fourth is blank. First is blank. So what must be the first one? The first box includes alphabets. Alphabets together forms the words. Words together form sentences. Sentences together can form the paragraph passages. The paragraphs together form the chapters, and all the chapters together forms the book. So we have seen the flow chart just now. So a book, how the book is written. So the things which are included are like alphabets, then words, then sentences, then paragraph, chapters, and then book is formed. So our body is also formed with the cells, then tissues, then organs, then organ systems, and finally the body. So cells form the basis of the structure and function of all living organisms. It is only with the help of cells the living organisms carry out all the different life processes. So in the figure 11.2 you can see a compound microscope. Measurement and observation of cells. In 1673, Anton van Leeuwenhoek assembled various lenses to construct a microscope. He was the first to observe live bacterial and protozoan cells under the microscope. So you all know what is microscope and when and for what the microscope is used. So in the figure 11.2 you can see a compound microscope. So do you know what is the use of microscope? Microscope is used to observe the microorganisms which we are not able to see with our naked eyes. So let's start with the measurement and observation of cells. In 1673, Anton van Leeuwenhoek assembled various lenses to construct a microscope. He was the first to observe live bacterial and protozoan cells under the microscope. Cells are extremely minute in size. They are very, very small in size. We cannot see that cells with our naked eyes. Micrometer and nanometer are the units used for measuring their sizes. The compound microscope is used for observing cells. An object on a glass slide magnified many times by the lenses of this microscope. So as there are units for like centimeter, kilometer, kilogram, so even the cells, to measure their size, the units are micrometer and nanometer. Okay, just observe the figure 11.4. Various cells. So these are the different types of cells. Their functions are also different. There is a great variation in shapes of cells. You can see the shapes are also different. The shapes are mainly related to their function. Observe the cells of different shapes shown below. Amoeba, red blood cells, that means RBCs, surface cells, muscle cells, a bone cell, a sperm, a nerve cell, neuron, an egg cell, a fat cell, spirogyra. These are different types of cells with different shapes, with different functions. Cells are of many different shapes. They are circular, they are rod shaped, they are spiral, they are rectangle, they are oval, they are column shaped. Each cell contains various components for carrying out the life processes of the living organisms. These components are called organelles. These organelles are studied in detail with the help of the electron microscope, which can magnify images up to 2 billion times their actual size. There are two main types of cells. animal cells and plant cells. These cells consist of various types of membrane-bound cell organelles. Plant cells have a definite shape due to presence of cell wall around. Besides, unlike animal cells, plant cells contain single large vacuole. All these cells are known as eukaryotic cells. Now, let us observe the plant cell. Endoplasmic reticulum, nucleus, nucleolus, ribosomes, cytoplasm, mitochondria, Golgi apparatus, cell membrane, that is plasma, chloroplast, vacuole, and cell wall. Now let's see the animal cell. What is included in it? Small vacuoles, lysosomes, Golgi vesicles, endoplasmic reticulum, cell membrane, plasma, mitochondria, Golgi apparatus, nucleus, centrosome, microtubulus, cytoplasm, and ribosomes. Let us study the differences now. Difference between plant cell and animal cell. Cell wall. Cell wall is present and is composed of cellulose in plant cell. In animal cell, cell wall is absent. Shape. Plant cell is rectangular and has a fixed shape. In animal cell is round and irregular shape. Vacuole. It has one large central vacuole taking up 90% of cell volume and is filled with watery fluid. In animal cell, it has a small vacuoles much smaller than plant cells. Chloroplast. Plant cells have chloroplast because they make their own food. Animal cells don't have chloroplast. Plastids. Plastids are present in plant cell. Plastids are absent in animal cells. Lysosomes. Lysosomes usually not evident. In animal cells, lysosomes occur in cytoplasm. Centrosome is absent in plant cell and centrosome is present that helps in cell division. Now, let us take a look at the similarities between the plant cell and animal cell. Endoplasmic reticulum. Endoplasmic reticulum is present in plant cell and is present in animal cell. Ribosomes are present in plant cell and also in animal cells. Mitochondria is present in plant cell as well as animal cell. Nucleus is present at periphery of the cell. Nucleus is centrally located in the animal cell, means nucleus is present in both the cells. Golgi apparatus is present in plant cells as well as in animal cells. Cytoplasm is not as dense as an animal cell. Cytoplasm is present and is dense in animal cell.